In this session, we will be looking at geometric isomerism. How do we see? Because to understand and appreciate geometric isomerism, we do need to know some biological process which are very crucial for us to see. There is a beautiful lake, the Dal Lake in Kashmir, Srinagar. So, how we actually see this particular beautiful image through our eyes? If you look at the vision, it is basically a biological process. So, the whole process is actually given here. Rhodopsin is an important protein which is involved in our vision, and the major transformation is the cis-retinal and trans-retinal. Two important compounds are involved in our vision. So, how do we actually see is based on a process, and in this process cis-retinal is one of the important compound which absorbs a photon, that is the light which falls on it, and it gets isomerized to trans-retinal, and this trans-retinal is actually helping us to see what we are actually seeing. So, this cis-trans isomerism is actually one of the important process for us to see. So, if we look at the isomers of retinal, the cis isomer is given here. So, we will be studying in this particular session what are cis isomer, what are trans isomers, and how to represent them. All these things we will be studying. So, before we start, let us look at how we actually can visually see the things. So, our vision basically requires a protein called rhodopsin. So, this rhodopsin has a small helper molecule, that is cis-11 retinal. So, this is the cis isomer, this is having an aldehyde group. So, the name given is retinal, and this is actually made from vitamin A. This cis-retinal on exposure to photons, that is light, is getting converted to all trans isomer, that is all trans retinal as shown here. This transformation is very essential for us to see. So, this is the importance of geometric isomerism in biological systems, and in this particular session we will be looking at what are cis isomers, what are trans isomers, and how do we represent them. All these things we will be studying. So, basically stereoisomerism is the topic we will be dealing now. This stereoisomerism is broadly classified into configurational isomerism and conformational isomerism. The later part of the series we will be studying about conformational isomerism in detail. In this particular session, we are looking at this configurational isomerism, which is broadly classified into geometric isomers and optical isomers. In this session, we will be studying about geometric isomerism, and in the next session, we will be studying about optical isomerism. So, in geometric isomerism, we have cis-trans isomers, and in the optical isomerism, we have diastereomers and enantiomers. So, what are geometric isomers? Basically, geometric isomers are molecules where certain atoms or groups are fixed in their spatial position with respect to one another, because in these kind of compounds, we have a fixed representation, or the molecule is fixed. Here the isomerism occurs due to restricted rotation across bonds. So, this restricted rotation may be due to presence of double bonds, or it may also be the presence of a ring structure. So, these two restrict the single bond rotations. Basically, the bond rotation is restricted by the presence of double bond or a ring structure. This leads to geometric isomers. The general nomenclature what we will be using are cis-trans isomers, EZ isomers, syn-anti isomers. We will look at all these three terms differently in this particular session. So, what are cis-trans isomers? Basically, the cis-trans isomers in double bond, we are going to look at only the first case, which is a double bond. So, two groups or atoms attached to separate atoms that are connected by a double bond, or they are contained in the ring. So, in this particular slide, we will be looking about the double bond cis-trans isomers. So, there is a double bond here represented, and there are two similar groups which are present on the same side. If you look at, these are present at the top, and there are also two other groups which are present at the bottom, which are also same, and there is a plane which is actually, when we are looking at across this plane, whether these two groups are on the same side of the plane. If it is the case, these kind of compounds are called cis isomers. That is, in the cis isomers, the each groups are lying on the same side of the plane. So, if it is same side, it is called cis isomer, and if the same groups are present in the opposite side of the plane. So, this is the central plane, and there is a group purple color at the top and one at the bottom. These are on the opposite side of this particular plane. So, if this type of arrangement is present, it is called trans isomer. And if you look at this compound here, the purple color group is present on the opposite side. So, should we call this as a trans isomer? But unfortunately, we cannot call this as a trans isomer, because if you look at the cis trans isomer, the two groups or atoms has to be attached to separate atoms that are connected by a double bond or present in a ring. So, that means here the two groups are present, the two purple atoms or groups are present on the same carbon atom. So, in these kind of cases, we do not have any cis trans isomers. So, we do not call these kind of compounds as cis trans isomer. So, to represent cis trans isomer, the same groups has to be present on either adjacent atoms, or it may also be far away atom in the case of cyclic systems. Then we call that as a cis isomer. If they are present on opposite side, they are called as trans isomers. When we talk about the ring, because we just saw about the double bond, if we are looking at the ring, rings also can show cis trans isomers. When we talk about ring, the two atoms or groups are said to be located cis to each other if they lie in the same side of the plane. So, they can be on the adjacent atom or far away atoms. When we talk about double bond, they have to be on the adjacent atom, but when it is a cyclic structure, they can also be from far away atoms. Say for example, here this is 1,2 substitution. As you know, the bond which is pointing upwards, that is the wedge bond, is both are on the same side, they are pointing upwards. So, that means this is called a cis arrangement. This can also be in a 1,3 position. If you start from here 1, 2, 3, so this is number 3 position. So, in this case we have 1, 2, 3 and 4. This is in the 4th carbon. So, this is a 1,4 cis arrangement. So, this is pointing upwards and the other group is also pointing upwards. So, when both are on the same side of the plane, this is called as cis arrangement. And when we go to the 1,2 arrangement, which is actually one is pointing up and this is pointing down. So, the wedge bond is pointing up and the broken bond is pointing downwards. So, they are in the opposite side. In those kind of cases, we call this as a 1,2 trans arrangement. Similarly, we have 1, 2, 3, this is 1,3 trans arrangement and this is 1, 2, 3, 4, this is 1,4 trans arrangement. So, this way we can represent cis and trans in a cyclic system, that is in rings, and the only difference between a double bond and the ring is, in the double bond we always look at adjacent atoms only. But in the case of cyclic system, it may be adjacent atom or it may also be far away atoms. So, this is how we represent cis trans isomers in cyclic systems.